Okay, this video is a tutorial for teaching you how to use the NCBI database tool to look for gene information. We'll focus this tutorial on looking for information about this gene HBA2 for humans on the NCBI website. You can click this link to directly go to the website, or you can always use Google to search for the NCBI website. We'll use the gene tool and type in HBA2 gene. Go search. Because we're looking for humans, we want the first search result, which indicates that this gene information is for human. Okay, the first question asks us the full name for this gene. This gene's full name is hemoglobin alpha 2. And it also asks us what is this gene located on. It's located on, if we look at the genomic context, it's on chromosome 16. If you're not sure what this 16 means, you can look at this picture here. It shows that HBA2 is here and it's located on chromosome 16. The third question asks whether this gene is a coding or non-coding gene, and next, how many exons and introns does this gene have? Let's go back to this information here. Now, this gene is a protein coding gene. And how many exons does this have? Under genomic context, we look for exon number, it's 3. So it's got 3 exons. If you are not sure, you can always scroll down to the next part, which is genomic region, transcripts, and products. Gives us picture of this gene. Now, this gene, the larger squares, these are 3 exons. They're intervening thinner lines. These thinner lines represent introns. Okay, the lighter green part are the untranslated region of the exons. Okay. Here we can see there are different numbers of different SNPs that are located on this gene. On the bottom here, these are the different CMV or copy number variations that exist in this gene. Okay, so what diseases are associated with mutation in this gene, and what is the function of this gene? Now, for diseases, we go to phenotypes. Okay, under phenotypes, we look for any associated health conditions. Here we see that alpha thalassemia, Heinz body anemias, and hemoglobin H disease are associated with this gene. Now, to answer the function of this gene, you have to go down to gene ontology. So general gene information. Gene ontology tells us the function that this gene has, the biological process this gene product is involved in, and whereabouts in the cell can we find this gene product. So function are all these. It tells us that it's an oxygen binding protein, iron binding protein, heme binding protein, and it's involved in process of oxygen transport. That makes sense because hemoglobin is a protein in the red blood cell, and it's responsible for binding to oxygen and transporting oxygen in your bloodstream. Okay, the next 3 questions asks us about the length of DNA in this gene, the length of messenger RNA, and the length of amino acids. And finally asks us if this gene has any conserved domains. If so, what is this conserved domain? Okay. So if we want to look for the length of this gene, we go down to sequence. Okay. We look for genomic sequence information. Let's go to GenBank link. Now GenBank tells us that this gene has 864 base pair. Okay, and we have all sorts of different information. If you scroll down to the bottom, this is the actual sequence of this gene. All these information you can also find in the previous page and also on the messenger RNA page and protein page. Let's go back to see the messenger RNA. Also under sequence information. Okay, sequence information, again under sequences, mRNA and proteins. This first link is the mRNA. The mRNA is about 122 base pairs. If you scroll down to the bottom here, it's going to tell you, okay, where the exons are. And what each exons represent. Okay, and also gives you the protein sequence. Let's look at the protein sequence. The protein sequence has about 142 amino acids. Okay, and if you scroll down to the bottom, this is the amino acid sequence. Now, if I want to look for conserved domain in this protein, okay, it's again under mRNA and protein, conserved domain. The number 1 shows us that there is one conserved domain in this protein, and this conserved domain is from amino acid 3 to amino acid 142 in this protein, and the domain is called HB alpha-like. If you want to see a clearer picture of this, you click on the summary, and you can see all the previous information, including the figure here. Okay, if there's an extra question asking you where the untranslated region is, if you want to find the untranslated region, you have to go back to the genomic GenBank link. And you would scroll down to the coding sequence here. Okay. The mRNA tells us where the exons are. So the first exon 1 to 161, the second exon 179 to 483, the third exon 626 to 864. Now, for the coding sequence means the translated region. It tells us that the translated region is 67 to 161. That means 1 to 66 is the 5' untranslated region, or 5' UTR. Let's look at the third exon, 626 to 754. That means 754 to the last base pair, 864. In this mRNA sequence, that's the untranslated region at the 3' end. So that's the 3' UTR. Okay, so this video kind of briefly tells you how to